Dear students, happy to meet you in this online mode of education. I am Dr.S.ANGELINA PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M. College for Women, Nagapattinam. The topic I am going to discuss with you today is about the mussel culture, which is coming under the paper Economic Zoology. Mussels are among the many invertebrates under the phylum Mollusca. Their wide distribution in the coastal areas of the Indo-Pacific region makes them the most easily gathered seafood organisms, contributing a significant percentage to the world marine bivalve production. In the wild, mussels are mostly found in the littoral zone, attached in clusters on various substrates. Being a filter feeder of phytoplankton and detritus, it is considered the most efficient converter of nutrients and organic matter produced by marine organisms in the aquatic environment into palatable and nutritious animal protein. Mussels were initially considered as a fouling organism by oyster growers. Mussel farming does not require highly sophisticated techniques compared to other aquaculture technologies. Even unskilled laborers, men, women, and miners can be employed in the preparation of spat collectors as well as harvesting. Locally available materials can be used, hence minimum capital investment is required. The mussel harvest can be marketed locally and with good prospects of export. Success in mussel farming, however, depends in providing some basic requirements to the bivalve, such as reasonable amount of sheltering of culture areas, good seawater quality, and sufficient food in the form of planktonic organisms. These pre-requirements are found in coastal waters or ideal sites for mussel cultivation. The biology of the mussel. The green mussel, Terna viridis, has separate sexes, although hermaphroditism usually occurs. Externally, it would be difficult to determine the sex. However, internally, the gonad tissue of a sexually matured male appears creamy white in color, while that of the female is reddish apricot. Sometimes, young sexually immature females cannot be distinguishable by color from the male specimens. Mussels have two relatively distinct phases in their life cycle: a free swimming planktonic or larval stage and a sessile adult stage. The free swimming larvae remains planktonic for 7 to 15 days depending upon the water temperature, food supply, and availability of settling materials. At about 2 to 5 weeks old, the larvae with a measurement of 0.25 to 0.3 mm seek a suitable substrate to settle on, and final metamorphosis take place, changing its internal organ structure to the adult form. The young spat then grow rapidly, and within 4 to 8 weeks after settlement, they measure 3 to 4 mm in shell length. Subsequent growth of the bivalve can be distinguished into shell and body growth. The shell length does not necessarily reflect the meat content. During spawning or food storage, internal energy reserves are consumed while the shell may continue to grow. Overall growth of the mussel as far as shell measurement is concerned is influenced by factors like temperature, salinity, food availability, disturbances, and competition for space. On the other hand, body growth is affected by the season, which primarily relate to sexual cycle and overcrowding to a certain extent. Let us see about the growth. Green mussel shows a rapid growth rate by length of 8 mm to 13 mm per month. Under average culture conditions, green mussel and brown mussel attain a length of 80 to 88 mm with 36.5 to 40 gram of weight. The formed mussels give a better meat yield compared to mussels from the natural bed. The average edible portion of the meat yield is 27.2% to 33.3% of the total weight. Growth by length and weight are probably the most important criteria for assessing the success of the culture system. The growth of muscle is influenced by a number of environmental factors such as water quality, food availability, settling density, water current, and tidal exposure. Of this, mussels such as Perna viridis and Perna indica forms the most dominant cultivable species. The Central Marine Fisheries Research Institute has developed eco-friendly techniques for mussel developed cultures. And mussels are known to be unisexual. The gonad of mature female can easily be distinguished by its bright orange-red color form and which is differentiated from the male with a creamy yellow color. Let us see something about the reproduction. Mussels attain sexual maturity in 2 months, 15 to 28 mm. Spawning period is prolonged, extending from January to September, with peak spawning during June to September. The 4 main stages in the reproductive cycle are spent or resting, developing, ripe, and spawning. Fertilization is external. After fertilization, it attains pedivillager within 15 to 35 days. Pedivillager attaches on the settlers with the help of byssus threads and metamorphose to spat. Spat settlement takes place from July to September and attains seeding size in September. Let us see something about forming techniques. Open sea and estuarine areas from strong wave action are suitable for forming. Clear seawater with rich plankton production is ideal for mussel culture. Moderate water current will bring the required planktonic food and will carry away the excessive buildup of pseudophysis and silt in the culture area. The water should have a salinity of 27 to 35 ppt and temperature of 26 degree to 32 degree centigrade. Site should be free from domestic, industrial, and sewage pollution. Let us see something about open sea forming. In open sea forming, the depth at the site should be above 5 m without strong wave action, less turbulent, and with high primary productivity. Long line and raft culture techniques are ideal for open sea forming. Mussels grown on long lines become smoothed by the naturally settling juvenile mussels and other fouling organisms. Effective utilization of easily available materials for fabrication of long line and raft can be done. Disadvantages of this forming are the poaching and unpredicted climate changes. Protected bays are ideal for mussel forming. We can see something about estuarine forming. Compared to sea, open sea and estuarine ecosystem with less turbulent and shallow depth with below 4 meter are suitable for mussel forming. Culture of mussels on horizontal ropes results in high productivity due to the effective utilization of the primary productivity. Rack culture is ideal for estuarine conditions. Fluctuation in salinity during monsoon season and pollution through domestic and industrial waste are the main constraints in estuarine mussel forming. Let us see something about culture aspects. The cultivation of mussels has taken various forms in different countries of the world. However, as in all forming procedures, it requires careful consideration of environmental, ecological, and seasonal factors in order to ensure proper growth and survival of the stock through harvest. Criteria for site selection include in prospecting sites for mussel cultivation, well protected or sheltered covers and bays are preferred than open unprotected areas. Sites affected by strong wind and big waves could damage the stock and culture materials and therefore must be avoided. Another important consideration is the presence of natural mussel spatfall. Areas serving as catchment basins for excessive floodwaters during heavy rains should not be selected. Floodwaters would instantly change the temperature and salinity of the seawater. Sites accessible by land or water transportation are preferred so that culture materials and harvest can be transported easily. Let us see something about water quality. Areas rich in plankton, usually greenish in color, should be selected. Water should be clean and free from pollution. Sites near densely populated areas should not be selected in order to avoid domestic pollution. In addition, the culture areas should be far from dumping activities of industrial waste and agricultural pesticides and herbicides. Waters are too rich in nutrients, which may cause dinoflagellate blooms and render the mussels temporarily dangerous for human consumption, causing either gastrointestinal troubles or sometimes paralytic poisoning should be avoided. Physicochemical parameters of waters are also important factors to be considered. The area selected should have a water temperature ranging from 27 to 30 degrees centigrade, which is the optimum range required for mussel growth. Water salinity may be around 27 to 35 ppt. When we see the culture methods, the bottom culture is one of the important methods. Here in this bottom culture, as the name implies, growing mussels directly on the bottom. In this culture system, a firm bottom is required with adequate tidal flow to prevent slit deposition, removal of excreta, and to provide sufficient oxygen for the cultured animals. This method requires a minimum investment. Disadvantages of this type of culture is the heavy predation by oyster grills, starfish, and crabs. Also, siltation, poor growth, and relatively low yields per unit culture area. Rack method. This method is suitable for estuaries and shallow bays. The racks are fabricated placing bamboo casuarina poles vertically and horizontally, tying and latching with nylon ropes. Bamboo or casuarina poles are driven into the bottom and spaced at a distance of 1 or 2 meters. These stakes are connected horizontally with the poles. The horizontal pole should be above the level of water at high tide, and seeded ropes are suspended from the same. Let us see something about deep water culture. It includes raft culture. Mussel raft culture has been practiced in Spain for a long time. Mussel seeds that settle freely on rocks or on rope collectors are suspended from a raft. When the weight of the bivalves on a given rope exceeds a certain limit, the rope is taken out and again distributed over a greater length until marketable size. It is a continuous thinning of the mussel stock to provide ample space to grow. Marketable shellfish are detached from the rope, purified in basins before marketing. The raft may be an old wooden boat with a system of outrigger built around it. Other kinds of rafts could be a catamaran type boat carrying about 1,000 rope hangings, or just an ordinary plain wooden raft with floats and anchors. Floats can be made of plastic, wood, and oil drums. The rafts are transferred from one place to another using a motor boat. Let us see about the raft culture. Here the advantages of this type culture are include reduced predation, utilization of planktonic food at all levels of water, and minimum siltation. In hanging method, the process starts with the preparation of the spat collectors. These collectors are hang on horizontal bamboo poles at 0.5 m. A piece of steel or stone is attached at the end of the rope to prevent the collector to float to the surface. Setting of collectors is timed with the spawning season of the mussels. Spats collected are allowed to grow on the collectors until marketable size. Other materials utilized as collectors are rubber sheets and strips from old tires. Harvested mussels are cleaned of the dirt and mud by dipping the collectors several times in the water. The process may be require more labors, but the ease in harvesting and availability of local materials for culture purposes makes it very adaptable under local conditions. Tray culture, here the mussels is limited to detached culture of mussels. Bamboo or metal trays with 1.5 meter with up to 15 meter sidings are used. The trays either hang between poles of the hanging or stake methods or suspended on 4 bamboo posts. Let us see something about wigwam culture. The wigwam method requires central bamboo pole serving as a pivot from which 8 full-length bamboo poles are made to radiate by firmly staking the butt ends into the bottom and nailing the ends to the central pole in a wigwam fashion. To further support the structure, horizontal bamboo braces are nailed to the outside frame above the low tide mark. Pads settle on the bamboos and are allowed to grow to the marketable size between 8 to 10 months. In rope web culture, here in this method, first tried in Sabian Bay, Capiz in 1975 by a private company. It is an expensive type of culture utilizing synthetic nylon ropes with 12 mm in diameter. The ropes are made into webs tied vertically to bamboo poles. A web consists of 2 parallel ropes with a length of 5 m and each positioned with 2 m apart. They are connected to each other by a 40 m long rope tied or fastened in a zigzag fashion at an interval of 40 centimeters between knots along each of the parallel rope. Bamboo pegs 20 centimeter in length and 1 centimeter width are inserted into the rope at 40 centimeter interval to prevent sliding of the crop as it grows bigger. In harvesting, the rope webs are united and the clusters of mussels are detached. In stake method, it is midway between the rack and bottom methods. Bamboo poles with 4 to 6 meter in length are stacked firmly at the bottom in rows 0.5 to 1 m apart during low tide in areas about 3 m deep and above. In areas where water content and also the water current is strong, bamboo poles are kept in place by nailing long horizontal bamboo supports between the rows. Since mussels need to be submerged at all times, it is not necessary that the tip of the poles protrude above the low water level after stacking. However, boundary poles should extend above the high water level. In stacking, enough space between plots is allowed for the passage of the farmer's banker during maintenance. Collected spuds are allowed to grow in situ until marketable size 5 to 10 centimeters after 6 to 10 months. It has been observed that about 2,000 to 3,000 seeds attached on 1 meter of stack with 1 to 2 meter below low water level. The mussels are harvested by pulling out the poles and bringing them ashore on a banker. Some poles may be reused during the next season. Let us see something about long line culture. Here, it is an alternative to raft culture in areas less protected from wave action. A long line supported by a series of small floats joined by a cable or chain and anchored at the bottom on both ends is employed. Collected mussel spuds on ropes or strings are suspended on the line. The structure is fairly flexible. In horizontal method, it is ideal in shallow areas with a minimum level of water column. Seeded rope were suspended by tying upward by ropes to horizontal poles, but both the ends will be stretched and tied in vertical poles erected in opposite sides in the form structure. In the estuaries of Malabar, most of the farmers are following this method. Let us see something about seed collection and seeding on rope. The site selected for collection of seeds should be free from pollutants. Seeds collected from the submerged areas will be healthier. After removing other organisms and weeds, the seeds were washed thoroughly in seawater. About 500 to 750 grams of seed is required for seeding on 1 meter length of rope. The ideal size of the seed is 15 to 25 mm length with 1 to 2 grams of weight. The length of the rope is decided by considering the depth where the raft or rag is positioned. Suspending the seeded rope on a rag, it must be tied in such a way that the upper seeded portion on the rope should not get exposed during the low tide. Nylon rope of 12 to 14 mm or 15 to 20 mm coir rope can be used for seeding. Old cotton net, cotton mosquito net, or cheap cotton cloth are used for covering the seeds around the rope. Cotton netting of required width and length is placed on the floor, and required quantity of seeds spread over the net one end to another end. The rope is kept above the net and is tightly stitched in such a way that the seeds spread uniformly around the rope. The cloth will disintegrate within 2 to 3 days. By this time, seeds will secrete byssus thread and will get attached itself to the rope. To avoid slipping of the mussels, knots are made on seeded rope at a distance of 25 centimeters. Placing split bamboo pegs in the ropes at regular intervals will also serve the purpose. When we see the growth phase, the seeds get attached to ropes and show faster growth in the suspended column water. If the seed is not uniformly attached, crowded position always shows slipping. To avoid slipping, periodical examination of seeded rope and thinning of the same is essential. The ropes also should be suspended in such a way that it will not touch the bottom, as well as the seeded portion is not exposed to longer period during low tide. Seeded mussels on the upper position of the rope shows faster growth due to the abundance of phytoplankton. For the better growth, the seeded ropes should be spaced at the distance of 25 centimeters. In open sea farming, growth of mussel is very rapid. They attain 80 to 110 mm in 5 to 6 months with an average growth of 13.5 mm per month and an average weight of 35 to 45 grams. This growth is observed in farms at various locations. In estuarine farming, mussels attain 75 to 90 mm in 5 months with an average weight of 35 to 40 grams and an average production of 10 to 12 kilograms per rope. Let us see about management. Constant vigil is required to see that the rack or raft is in position. Thinning may be done if necessary to avoid loss of mussel and to provide enough growing space. Periodic removal of fouling organisms like barnacles, polychaetes, and ascidians is to be done for improved growth. Diseases and poisoning is very important. Mussels are said to be harmful when consumed during periods of red tide. This mainly occurs due to algal bloom with the help of dinoflagellates bloom or diatoms or with cyanobacteria. They will produce potent toxins that can find their way through the food chain or human, causing a variety of gastrointestinal and neurological illness such as paralytic shellfish poisoning, diarrhea, also neurotoxin shellfish poisoning. Another new toxin identified is exotoxin, which affects the nervous system. We have to know about the harvesting procedure. Harvesters should be aware of the stress caused during the harvesting process. In harvesting mussels, special care is needed. Pulling them or using a dull scraper may tear the byssus thread. This will result in loss of moisture after harvest or cause physical damage, causing early death of the bivalve. The right procedure is to cut the byssus thread and leave it intact to the body. Exposure to sun, bagging and transport also increases the stress of the mussels. The marketing, depurated mussels are then mainly sold through local markets as a live shell on mussel. At present, processing units use only a small quantity of cultured mussel. New strategies may need to be developed to fully exploit the domestic market. Meat from depurated mussel can be struck in first fresh condition or after boiling or steaming. Further processing of the mussel meat can be done after blanching in 5% salt solution for 5 minutes. Harvest will be done when the mussels reach marketable size and condition index is high, that is before the spawning and onset of monsoon. Normally, harvest season is from April to June. Mussel rope is collected manually and brought to the shore for harvest and washed thoroughly using jet wash to remove grit and silt. The mussels are separated from the ropes are maintained in recirculating seawater for 24 hours and washed again in fresh water. And also this method of depuration is effective in reducing the bacterial load of the mussel meat. To conclude, mussel culture has played greater role in meeting the increasing protein demands of the human population. Bivalves such as oyster, mussel and palm are the most important cultivable organisms all over the world. Thank you.